Okay, moving on. Give him the pointer. There he is. Moving on to the rest of chapter 1. So this rest of this chapter deals with scientific method, and the scientific method is so crucial. One thing I want you to appreciate, science is a process. It's not wham bam, that's what it is. We have to go through this process using scientific method to get a better understanding of things, why things are happening. Sometimes it takes multiple experiments and time to figure out why something is the way it is. We're seeing this really play out with this current pandemic and our understanding of the virus. We're understanding more about it, how to treat it. There is no real cure. I know there's a big debate about that. I've seen friends totally lose friendships over years over the debate about that. But the scientific method is super important. I want to first ask, when someone says they're a scientist, what do you think? Do you think, probably Albert Einstein? Someone that's a really smart genius, probably a nerd, sits in there reading books all the time. You could probably think of a mad scientist in the lab concocting some crazy things, making some weird half this, half that. You probably think of someone in a white lab coat, doing stuff in a very white sterile room. One thing I do want to point it out is how they show it in TV and movies is like two totally different worlds. And this cartoon just sums it up for me. Movie scientist versus real scientist. It's not as quick and easy as the TV shows or movies will show it. It takes time. If you ever want to really piss off a scientist, go watch a movie like Contagion with them, and they'll point out the flaws, and it's like, okay, that's not realistic, and all the other stuff. Or the CSI shows, it's like, oh, I put this in my trunk and it tells me exactly you're the killer. Boom, boom, boom. It didn't do that. But I want you to appreciate the scientific process a little bit more after today. So the scientific methods is a series of steps. But as I mentioned, science isn't just memorizing facts. We want to find a better understanding of the unknown. We want to understand, why is this happening? Why? Things are always changing, and we want to get to the foundation of why that changing is happening. What's driving it? One way to tell if someone's a scientist, they ask why a lot. Or a kid. If you have a kid, ask like why. Why? A lot of kids do that, but if they excessively ask why is this happening? Why is that? That's just the process. That's how their brain is just going through stuff. Scientists love to do that. We ask why. Okay, why is it doing that? Why is my plant doing that? Why are you suddenly like crapping out and dying on my balcony plant? We ask why. And with science, one thing I want to do point out is it's not cut and dry. We have to consider all possible reasons why something is happening. And this can make research super frustrating because you might have thought you had it all figured out. You've done your experiments, your data is beautiful, your graphs look great. You go present at a conference and then someone comes and it's like, well, did you think about this? And then you're like, well, crud, no, I didn't. And it could totally put a flaw into there. So research is a very, very stressful field. I'll talk about that a little bit later. Now, you use the scientific method all the time. You don't realize it, but you do. An example I'll kind of talk about is your phone. You misplace your phone. They're attached to us. Mine's right in front of me. You use it all the time. So you're going there and think about, oh, maybe I need to call my mom or something like that. So first thing you do is you observe that it's missing. You go to reach for it and it's not there. All right, so you make an observation. Next thing you do is you're going to make a hypothesis or an educated guess, and it's like, well, I left it somewhere. It's in another place, okay? So then you're going to come up with an experiment. How are you going to figure out where your phone is? So you kind of go through your brain and think about, where are all the places I've been and where might have I used it last? So maybe it's in here, maybe it's in that room. So that you come up and design the experiment, and then you actually go and conduct the experiment. So you get up. If you let it in the bathroom, did you find it? It's not in the bathroom, is it in the kitchen? Did you find it? So you keep repeating that experiment until you actually find it. And then once you find it, or hopefully not, you totally lost it, you come up with your conclusions. This process, looks a lot more complicated, but it's simple. You do it all the time. Now, there's different levels when it comes to parts of the scientific method. One of the key things with the scientific method is you're coming up with a hypothesis, or it's a question that you're going to be testing. So one thing with hypotheses, you need to make sure they're very tentative, and you're trying to answer this question. And when you go through and design your experiments, you need to design them so they're either going to support or reject your hypothesis. And they have to be testable. In science, we have to be able to measure things, weight, length, time, something that has like a unit that we can put behind it. There are certain things that we can't measure. I put psychic abilities here, the paranormal shows. We love to watch them, but how do we actually measure those? It's kind of interesting to watch these kind of things. When you come with that, you can come up with different types of hypotheses. You'll hear the term null or alternative. They're just talking about the observed effect, if you will or will not see a change. Now, after something is tested over and over again, it might advance to the next level, which is called a theory. It's a prediction of why something's happening. So multiple experiments to determine why something is. Theory of evolution is a good one. We'll talk about it. Now, sometimes theories can change. When we get more increases in technology, it might alter what we know about it. Now, the next level up is law. Law is exactly why something happens, and it's going to be backed up by math is the key thing. Law of relativity, law of gravity. Not many things get to this level right here. Most of the time, they'll stop after theory. Now, this is a short little YouTube video that kind of explains each of these in a little bit more detail to give you a little bit more information. Now, experimental design is key, like I say right there. You want to clearly be able to answer your hypothesis, and if you don't clearly design your experiment, it can lead to a lot of problems. Now, it may require multiple experiments to answer your overall hypothesis, and the one thing, it has to be repeatable. Now, when I say repeatable, that means you can do it and get the same results, and someone else can do it and get the same results. Now, just because you said it works in your hands, that's great. But if it doesn't work for someone else, means there's probably some flaws in your experiment. I'll talk about this, and maybe you'll have a link to it later on, is, sometimes if you say, oh, here's this great new technique and it's going to revolutionize the field, and then when other people try it and it doesn't work, it can lead to problems. We've had this in the stem cell field. We saw this in 2014. The reprogramming of stem cells takes time. It takes a lot of effort and time and work. There was this article that came out and said, hey, we can cut this in half by using this certain type of acid. Well, people are like, well, crud, that's great. It changes the field. Well, people started trying to do that technique in a lab. They cannot get it to work. Wound up that the method was falsified. They were not able to actually do what they could. They falsified some of their data, and they had to retract the story. And that can lead to very, very bad things. So here's a cool link to an article that talks about that. Now, in your experiments, you can have one or more variables. There's two key terms right here. Basically, a variable is a factor or trait or condition that you're going to measure. So, number of barnacles, height of the plant. You can have independent or dependent variables. Now, the independent is what you're going to alter, you're going to manipulate. The dependent is what you're going to measure. So, it's going to depend on what you do, and you're going to measure changes in that. So normally when you look at a graph, the dependent variable is going to show up on the y-axis, and your independent will be on the x-axis. Now, with your experiments, you make sure you set good controls. There'll be positive and negative controls. When you do the virtual lab and macromolecules, I think you might talk about it a little bit. Oh, no, you'll hit them first in the scientific method virtual lab. So I do have a little bit more information about controls in there. Now, with science, one of the things you really want to control is bias. All right, so bias, what it does is it influences your results, because you want things to work so bad. You want your results to go great so you can make this great paper and get published, and maybe if it's a certain drug, you'll maybe get some benefits and make money off it, all kinds of things. Now, the bias can actually influenced by the person measuring or the person taking it. You might have heard of the placebo effect. So when clinical drug trials, what they'll do is they'll have a control group, which gets a placebo. It's like a sugar pill. And sometimes when people think that they don't know if they're getting it or not, and they just think, okay, I'm getting this drug and it's making me better. And they can think they're getting better, but the actual physical conditions are not. So we want to eliminate bias in all our experiments. So we do these called blind studies and double-blind studies. So basically, a blind study is just limiting the participant. So the placebo effect is a great one. So the people that are in the clinical trial have no idea if they're getting the actual drug or just placebo. So the vaccine trials probably are going out right now, have this group. They'll have a group that's probably getting the actual vaccine and a group that's not. So we can actually see, a change between with or without and what the results are. The people that are getting it have no idea. Now, a double-blind study also limits the researcher's knowledge and the participant groups. These are the most ethical because, the researcher might kind of be skewed in how they view things. They might write the results a little bit more, it's like, okay, no, let me round that up. With a double-blind study, there's like someone totally independent that's going to make all the measurements, and then the data is sent then to be analyzed by the researcher. They have no idea and no part in making the measurements. I kind of talk about this. Your project is subject to ugly baby syndrome. By that means it's yours. You think it's the greatest thing in the world, but what these blind studies and double-blind studies is it kind of eliminates that, and it kind of makes you aware. It's like, oh, maybe this isn't as great as it used to be, and it will point out some of the flaws that you have. You need all these great things. You have all these graphs. You have all this data. Now you got to push. Now, this is so key right now. The peer review process is very important for science. We want to share our results with each other. What's the point of doing all this work if we can't share it with other people? Now, what the peer review process is, it's going to eliminate bad science. So studies that are flawed, they're biased, will be eliminated thanks to the peer review process. So I wrote right here, it can be difficult. So what happens is you have this paper and you write it up and you decide you want to go to a journal like Science or Nature. What you do is you submit your paper, and then they're going to have an initial look at it and say, okay, it meets the standards, maybe not for this journal, and you get a quick yes or no. If you get a no, then you just pick a journal and you keep going until someone says, maybe we'll consider it. If you get the maybe, we'll consider it, next thing they do is they send your paper off to 3 different scientists. They're independent reviewers. You have no idea who they are. What they do is they read your paper and pretty much rip it apart. They look for all the flaws in your study. So what they do is find, okay, this is great. Sometimes we'll have a person say, you need to do like all this over again. This experiment's great, but have you thought about this? What they do is they find the bad science and maybe say, okay, you're almost there, but have you thought about this? And then what they'll do is they'll say a hard yes or no. And sometimes if it's a complete no, then there's nothing you can do about it. But if they say yes, but you might have to modify and do experiments to please them. So you go through, make the changes, and submit it again, and you can go through this process over and over again. Eventually, if everyone's happy, once the reviewers are happy, you get submitted into the journal. This is going to be a primary source, so your Science, your Nature's, your Cell, your peer-reviewed journals. Peer-reviewed articles are going to be very easy and hard to read. You might have some associated to your specific field. I have links to some in the slides that you can look at and see like a primary versus a secondary source. But primary sources are where we're going to get the most accurate data. Now, a secondary source can be different types. There can be books, news reports, pretty much anything you see on the internet, advertisements. What a secondary source is, it takes a primary source and it kind of simplifies it and makes it to where the common person can understand it. They're not dumbing it down. They're just pulling out the key points. And this is why this is important. This is why it's important to you, and it might change your life. And, yay, science. Now, with secondary sources, be skeptical because, they're only cherry picking what they want you to see. My advice, it sounds too good to be true, go back to the primary. Another thing to really be careful is anecdotal evidence. We're seeing this like crazy right now. This is based on what a person experienced. Yeah, your patients might have worked great using this one drug, but when another hospital used it and did similar things, it didn't work for them. Or, you grandma might have a certain medicine that combination she wants you to take, like drink the hot water with 2 tablespoons of honey at this temperature, and it'll make you feel better. It's all based on experience. If you want your stuff to be, legit, you gotta have the experimental data. Be kind of skeptical about this anecdotal evidence. Always go to the primary if you're doubting something. So science in the news can be very, very, very scary, and this pandemic has really been scary because I think science has really gotten the black eye because a lot of people are doubting it. Secondary sources, I mentioned it here, may be missing critical information, or they might report it incorrectly. They might not be reading it. A lot of times retractions are reported. This is really big with the anti-vax movement. A lot of people are like, oh, it causes autism because it's one paper. Well, that paper is actually retracted. Pay attention to stuff like this. Consider the source of your media reports. This is so big right now because people don't know who to trust. Go to like a neutral source. There's like a cool neat graphic that tells you like how biased different stations are. I always go to different ones. I check multiple stations, what they're reporting, and I always go to the primary, as I keep mentioning. Be super careful with the internet because anyone could post anything. Don't get your information solely from social media. I think a lot of people do, and that's led to a lot of problems these days. If you are using social media to solely get your news, please explore a little bit more because share something and false information gets shared, and the next thing you know, it goes viral, and we just do stupid things. We really do. The human population does a lot of stupid stuff, and we do it to ourselves. Hopefully through this, you're going to understand the process of science and think of it a little bit more critically. Your very first discussion post talks a little bit about, because the term fake news really took off since this last election, and what's true and what's not. So what you're going to do in the first discussion post is you're actually going to look for something like a primary article or something that seems too good to be true. Like when you hear it on the news, like 1 hour, 1 glass of red wine equals 1 hour of cardio at the gym. Really? Because I just did about 2 hours last night. So come on. So you need to go look at articles and actually research it a little bit. So you'll go through this process a little bit of this course. So this is just to kind of show you the difference between a primary and a secondary source here at the top. So this is talking about autism because people want to know what causes it. So Science Daily is a great secondary source. What they do is they take all these primary sources and they kind of put them into short little snippets. I go here maybe like once every other day. Or not once every day, but every other day pretty much, at least once a week. And just to kind of get an overview of what's going on. You click on the article and it'll kind of sum up what the primary is, and then it actually has a link to the primary most of the time at the bottom. This bottom link is the actual primary article, and these are hard to read. Reading a scientific paper is difficult. So, for more information, go to the primary, but there are some good secondary sites out there. I think I give you some links to them in one of your discussion posts to actually look at. Now, one of the things I do want to talk about is retractions. I didn't mention them earlier. This is a link to talking about some different retractions that have happened in science. The peer review process is supposed to catch the bad stuff, but sometimes it does slide through the gaps. So here's some information about some that have happened in the past. So just a little bit more information for you. And I think the last thing I have is how to read scientific papers. So this is just showing you the anatomy. If you want to go to that primary paper and read this, good for you. One thing I will admit is, especially when I was doing my lit review for my PhD research, I didn't read all the papers from start to finish. I actually read them in different parts. The abstract is like a short little summary. Most of the time I'll read that first to see if it even relates to what I'm kind of researching on. Introductions give you like a background, like why is it important. Sometimes, you'll see the introductions on a certain topic get kind of repetitive, but it's going to cite why their work is important, what gap in knowledge you're trying to fill. Sometimes I skip straight to the results and actually look at the figures. Figures should stand by themselves. They should tell a whole story on their own and should not have to rely on a lot of word text within the paper to explain why it's important. Discussion post is just our discussion part is just going to tell you why they think it is what it is. The methods, if you're actually going to redo it yourself, and then the references, you'll see hundreds of different references when it comes to scientific papers. But don't get bogged down. Like I said, there's different parts. I don't read it from start to finish. Sometimes I'll look at results, I'll read the abstract, then maybe I'll go look at discussion. There's all kinds of different parts to it. So I think that's the last one. Yes. So right here, this is some review videos about the scientific method in general. You're not required to watch these, but these are here to help you. It's more like, hey, here's something else. Sometimes listening to someone else talk about it helps a little bit too. I'm going to stop that, and that wraps up chapter 1.